Hello friends, welcome back to my YouTube channel Nastin Padasali. So in this video, I'm going to tell you list of questions that you can challenge with regarding to GATE biotechnology paper. At the same time, I'm going to tell you list of questions that you should not challenge. Okay, because for challenging each question, you need to pay an amount of 500 rupees. Okay, so you can input your ideas in the comment section, and if you are really in a plan of challenging that particular question, means just discuss us in the comment section. Just get ideas from any student like you, and then go for challenge. Okay. So first question. So this is question number 17. Dendritic cells are involved in cross-presentation of antigen. Which of the following proteins are required for cross-presentation? This is a MCQ type of question. And in the answer key, they just mention option A is correct, like that they just mentioned. Which means only basic leucine zipper ATF like transcription factor 3 are involved in the or required. It's a required protein for cross-presentation. Like that they had given. This the answer A is correct, like that they had given the official answer key. So for this question, you should not challenge, only answer A is alone correct. I could able to find the research article where they are discussing the role of this BAT3 in cross presentation of dendritic cell, because some YouTube channel where they had discussed the answer key for GATE BT, they just mention option B is correct, which means P, that is basic leucine zipper ATF like transcription factor 3, and solute carrier family 10 member 1 are involved, like that they just mentioned. Only option P alone is correct. This solute carrier family 10 membrane 1 is involved in a digestion process. It is a co-transporter which is involving in reabsorption of bile salt by co-transporting sodium and bile salt across the membrane. Okay, so for this question, option A is correct. So don't challenge this particular question. Okay. So second questions, which is literally confusing or whatever it might be. So that is question number 28. Which one of the following methods cannot be used to determine the secondary structure content of protein? So this is a MCQ type of question, and in the official answer key they just mention option C, that is mass spectroscopy cannot be used to determine the secondary structure of protein, like that they had given. But according to me, for this question, option D is correct. So we all know X-ray crystallography and NMR are employed for determining the tertiary structure of protein. But here they are asking which of the following cannot be used to determine the secondary structure of protein. And the correct answer is that is X-ray crystallography is the correct answer. But I don't know why they had mentioned mass spectroscopy. I had also quoted you this particular research article which has been published in the year 2013, where they just in the title itself they had clearly mentioned analysis of secondary structure in protein by chemical cross-linking coupled to mass spectroscopy. So chemical cross-linking mass spectroscopy has been employed in analyzing the secondary structure. So in the conclusion, you can be able to find this particular point. Information obtained by the cross-linking hold greatest promise for determining the secondary structure within a protein sequences through the use of all the analytical advantages of mass spectrometric detection. Okay, so for this question, option D is correct. X-ray crystallography is a technique which we can't able to use to determine the secondary structure of protein, whereas mass spectroscopy can be employed for determining the secondary structure of protein. So you can challenge this particular question. Next, the third question is question number 43. Which of the following is our plant protoplast fusogenic agent or agent? So this is a MSQ type of question, and for this particular question, official answer key they just mention option A, option B, option C, which means sodium nitrate, polyvinyl alcohol, polyethylene glycol are used, and it is a correct option. Okay, so don't need to challenge this particular don't need to challenge the answer for this particular question, because the same YouTube channel which has provided the answer key, there they just mention only option B and option C are correct. Okay, but when I analyze the Satya Narayan book for biotechnology, they just mention sodium nitrate and polyvinyl alcohol and polyethylene glycol, and sometimes calcium and magnesium ions can also be employed as a plant protoplast fusogenic agent. So don't challenge this particular question. So fourth question is direct DNA transfer method or methods used for plant genetic engineering is or are. And again, this is a MSQ type of question. For this particular question, option A, option B, option C are correct. So in my channel, just mention only option A and option B alone is correct. But according to the official answer key, option A, option B, option C are correct. So both the microparticle bombardment and electroporation are physical method, and polyethylene glycol is a chemical method. So here this polyethylene glycol is used as an alternative for biologistic delivery and is currently employed for your plastic transformation. Okay, so please don't challenge this particular question. It has been correctly given. Okay, so next is question number 56. For this particular question, many are telling the official answer key. They just mentioned the correct answer for this question is 2.1 to 2.3 milligram, like the judges mentioned from IIT Kanpur. But many students are telling the correct answer for this particular question is 1.1 mg. Okay, so yeah, if you want to challenge this question number 56 means you can challenge, or if you have any statement stating that 2.1 to 2.3 mg is correct means you can do tell me in the comment section, because I don't know how to solve this question. You can help me if you know the correct answer, or if you already solved means please do share us. Okay, so the next with regarding to question number 20. So here the question number itself mentioned wrong. Okay, actually it should be mentioned as question number 30, but here they had given us question number 20. When I discussed with my student, they just mentioned it is a typing mistake, like that they just mentioned, but I don't know whether they'll be giving marks for all like that. Okay, and next important thing, that might be the only reason again for this particular disease, Fabry disease in human is an X-linked disease, and they ask the probability in percentage for phenotypically normal father and a carrier mother to have son with the Fabry disease is that. Okay, according to official answer key, they just mentioned 25% is a probability, like that they just mentioned. But when we worked, and also in many YouTube channel they had provided a solved video for this particular question, they just mentioned the probability is 50%, and for me also 50% is the correct answer. So if you are interested, you can you can challenge this particular question number 30, which has been typed as a wrong question number 20 in the question paper. Okay, so you can challenge this particular question. So in GATE biotechnology, this question number 28, you can challenge, and next thing is with regarding to question number 56, you can challenge, and next question number 20, you can challenge. Okay, so these three questions you can challenge. Okay, this question number 56 is a 2-mark question. Question number 20, that is 30 question, is the it is a 1-mark question. Okay, so if you want to challenge, you can challenge, and if you have some other questions which you are doubting, if you are confusing means, please do tell me in the comment section so that we can discuss whether we can take the question for challenging or not. Okay, so thank you friends, thanks for watching this video.